Next topic, detection and measurement of radioisotopes. Radioisotopes can be detected and measured using various methods such as autoradiography, scintillation counting, and gamma counting. Autoradiography involves exposing a photographic film to the radioactive sample, which results in a pattern of silver grains that can be visualized under a microscope. Scintillation counting involves adding a liquid scintillator to the radioactive sample, which emits light when it interacts with the radiation emitted by the radioisotope. This light can be detected and measured using a photomultiplier tube. Then gamma counting involves using a gamma counter to detect gamma rays emitted by the radioisotope. Then incorporation of radioisotope. Radioisotopes can be incorporated into biological tissues and cells using different methods such as injection, inhalation, ingestion, and uptake by cells. The choice of method depends on the type of tissue or cell being studied and the desired outcome of the experiment. Then molecular imaging. Radiolabeling can be used for molecular imaging of biological process. This involves the use of radio tracers, which are compounds that incorporate a radioisotope and bind to specific molecules in the body. By tracking the movement of radio tracers using imaging techniques such as positron emission tomography or PET scan and single photon emission computed tomography or SPECT, researchers can visualize the molecular and cellular processes in vivo. Safety guidelines. The use of radioisotopes in biological research requires adherence to strictly followed safety guidelines to protect researchers and the environment from the harmful effects of radiation exposure. The guidelines include the use of protective clothing and the use of designated radioactive waste disposal containers and regular monitoring of radiation exposure levels. Radiolabeling techniques have many applications in biology, including studying metabolic pathways, gene expression, and protein interactions. These techniques are also used in medical research for diagnostic and therapeutic purposes, such as in PET scans and radiation therapy for cancer. Radiolabeling provides a sensitive and specific way to detect and measure biological molecules and can provide insights into the molecular and cellular mechanisms of biological processes. Advantages and disadvantages. One advantage of radiolabeling technique is their sensitivity and specificity, which allows for the detection and measurement of small amounts of biological molecules. However, the use of radioisotopes can also pose health risks to researchers and the environment and requires strict safety precautions. Additionally, the use of radiolabeling techniques can be expensive and time-consuming and may require specialized equipment and facilities. In summary, radiolabeling techniques are powerful tools for studying biological molecules and processes and have many applications in research and medicine. While there are risks and challenges associated with the use of radioisotopes, strict safety guidelines can mitigate these risks and allow researchers to use radiolabeling techniques safely and effectively. Then microscopic techniques. Utilization and of this microscopic techniques we are going to learn. So visualization of cells and subcellular components by light microscopy, resolving powers of different microscopes, all that's the topic we will be learning. So microscopy is an essential tool in biology that allows visualization of biological specimens at different scales, from whole organisms to subcellular structures. And microscopy techniques can provide important information about the structure and function of biological systems. So, light microscopy. Light microscopy uses visible light to magnify and resolve the image of a biological specimen. This technique allows visualization of the structure of cells and tissues, including organelles such as nuclei, mitochondria, and chloroplasts. It is commonly used in medical research and diagnostic purpose, for example, to study the morphology of tissues or to identify parasites in blood smears. One of the most commonly used light microscopy technique is the confocal microscopy, which uses laser light to scan the specimen and create a 3D image. One of the advantages of light microscopy is that it allows imaging of living cells and tissues. However, the resolution of light microscopy is limited by the diffraction of light, which makes it impossible to resolve structures smaller than 200 nm. Then electron microscopy. Electron microscopy or EM uses beams of electrons instead of light to image biological specimens. EM has a much higher resolution than light microscopy, making it possible to visualize structures as small as 0.1 nm. EM is used to visualize subcellular structures such as the structure of membranes and macromolecular complexes. There are two types of EM, like transmission electron microscopy TEM and scanning electron microscopy SEM. TEM is used to visualize thin sections of specimens, while SEM is used to visualize the surface of specimens. One of the disadvantages of EM is that specimens must be fixed and dehydrated, which can affect their structure and may introduce artifacts. Then fluorescence microscopy. Fluorescence microscopy is a type of light microscopy that uses fluorescent probes to label specific molecules or structures. This technique allows the visualization of specific molecules or structures within the cell, such as the localization of proteins within a cell or the tracking of a movement of specific molecules in living cells. Fluorescence microscopy can also be used in combination with other techniques such as confocal microscopy to create 3D images of specimens. One of the disadvantages of fluorescence microscopy is that the fluorescent probes can interfere with the function of the molecules they label, and the technique requires specialized equipment. Then freeze fracture microscopy. Freeze fracture microscopy is a type of EM that is used to visualize the internal structure of biological membranes. The specimen is rapidly frozen and then fractured, revealing the inner structure of the membrane. This technique is useful for studying the structure and function of biological membranes, such as the plasma membrane or the membranes of organelles. One of the advantages of freeze fracture microscopy is that it does not require staining, which can introduce artifacts. Then image processing. Image processing is a set of techniques used to enhance and analyze images obtained by microscopy. Image processing can be used to improve the contrast and resolution of images, to remove noise and artifacts, and to quantify the properties of biological structures. Image processing is essential for many microscopy techniques such as confocal microscopy, where the 3D images restructured and reconstructed from a series of 2D images. In conclusion, microscopy techniques are essential tools in biology that allow visualization of biological structures and processes. Different microscopy techniques have different advantages and disadvantages, and their applications depend on the specific biological question being addressed. Advances in microscopy techniques, such as the development of sugar resolution microscopy, have greatly expanded the scope of biological research. Next, electrophysiological methods. Electrophysiological methods involve the measurement of electrical activity in biological systems. These methods are widely used in neuroscience and cardiology to study the function of neurons and the heart, respectively. The following are some of the major electrophysiological methods used in biological research, like single neuron recording. In this method, the microelectrode is inserted into a single neuron to record its electrical activity. This method is useful for studying the properties of individual neurons, such as their firing rates and patterns. Next, patch clamp recording. This technique allows for the measurement of ion currents flowing through ion channels in the movement of a cell. In the membrane of a cell, it involves creating a seal between a patch of membrane and the tip of a microelectrode, allowing for the measurement of ion currents with high precision. Then electrocardiography, ECG. This method measures the electrical activity of the heart using electrodes placed on the skin. ECG is widely used for diagnosing heart conditions such as arrhythmias. Brain activity recording, electroencephalography and magnetoencephalography are used to ensure and measure the electrical and magnetic activity of the brain, respectively. These methods are useful for studying brain function, such as during different stages of sleep or in response to sensory stimuli. Then lesion and stimulation of the brain. Electrical stimulation or lesioning of specific brain regions can help identify the function of those regions in behavior and cognition. Then pharmacological testing. Electrophysiological methods can be used to study the effects of drugs on ion channels, neurons, and the heart. This helps in drug discovery and development. Then positron emission tomography, PET scan, magnetic resonance imaging, MRI, and functional MRI or fMRI, and computed axial tomography are non-invasive methods used to study brain function and diagnose diseases. Then electrophysiological methods have many advantages, including their high temporal resolution and high sensitivity and ability to study biological systems in real time. However, these methods also have some disadvantages, such as their invasive nature and the requirement for specialized equipment and expertise. Nonetheless, electrophysiological methods remain important tools for understanding the electrical activity of biological systems. So, methods in field biology. Next, methods in field biology involve the study of living organisms and their interactions in natural environment. Some of the important methods used in field biology are methods of estimating population density. Like population density is the number of individuals per unit area or volume. Methods for estimating population density of animals and plants include direct observation, mark and recapture, transect sampling, and quadrant sampling. Direct observation involves visually counting the individuals in a defined area, while mark and recapture involves capturing and marking a subset of individuals, releasing them back into the population, and then recapturing them at a later time to estimate the size of the population. Transect sampling involves sampling along a line or path, and quadrant sampling involves sampling within a defined area. Ranging patterns through direct, indirect, and remote observations. Ranging patterns refer to the movement and activity of animals in their natural habitat. Direct observation involves visually tracking the animals, while indirect observation involves tracking signs of their presence, such as footprints, droppings, or feeding behavior. Remote observation involves using technology such as radio collars, GPS, or camera traps to track the animals from a distance. Then, sampling methods in the study of behavior. Behavioral studies involve observing and measuring the behavior of animals in their natural habitat. Sampling methods include focal animal sampling, scan sampling, and instantaneous sampling. Focal animal sampling involves observing and recording the behavior of a single individual for a defined period of time. Scan sampling involves recording the behavior of all individuals in a group at definite intervals. While instantaneous sampling involves recording the behavior of all individuals at a specific point in time. Then next topic, habitat characterization. Habitat characterization involves describing and quantifying the physical and biological characteristics of an ecosystem. Ground-based methods include soil sampling, vegetation surveys, and water quality measurements. Remote sensing methods involve using satellite or aerial imagery to map the distribution and characteristics of vegetation, land cover, and water bodies. Field biology is important for understanding the ecology and behavior of organisms in their natural environment. It can inform conservation and management strategies for threatened or endangered species and can provide insights into the impacts of human activity on ecosystems. An optimistic message about a bright future. So, despite the challenges we face, there are many reasons to be optimistic about the future. So, that's it. Now, we are going to explore some very important topics in this exam, NET JRF exam of life science. So, let's explore those topics. First topic, developmental biology concept like autonomous and conditional specification. So, developmental biology is the study of how organisms grow and develop from a single cell into a complex organism with specialized cells and tissues. Developed from a single cell into a complex organism with specialized cells and tissues. Two key concepts in developmental biology are autonomous specification and conditional specification. Autonomous specification refers to the process by which cells in the developing embryo become different from each other due to intrinsic factors within each cell, such as differences in gene expression. This means that cells can differentiate into specific cell types even when isolated from other cells or tissues. But conditional specification, on the other hand, refers to the process by which cells become different from each other due to extrinsic factors in their environment, such as signals from nearby cells or the physical properties of their surroundings. In other words, cells can differentiate into specific cell types only when they receive specific signals from other cells or tissues. So, autonomous specification. During early embryonic development, cells start out as totipotent or pluripotent stem cells which have the ability to become any cell type in the body. As cells divide and differentiate, they become more restricted in their developmental potential and take on specific fates. In some cases, cells become specialized based on intrinsic factors such as differences in gene expression. For example, in the early embryo of many animals, the cells in the developing gut tube become specialized into different cell types depending on their position along the anterior-posterior axis. Cells at the anterior end of the gut tube express different genes and become different cell types than cells at the posterior end of the gut tube. Another example is the development of the nervous system. In the early embryo, a group of cells called the neural plate forms along the dorsal midline. These cells are specialized to become neurons and ganglia based on the expression of certain genes. Then conditional specification. Conditional specification occurs when cells become specialized in response to signals from their environment. And classic example of conditional specification is the development of mesoderm in the early embryo of the fruit fly Drosophila. The mesoderm gives rise to a variety of cell types, including muscle, heart, and blood cells. In Drosophila, the mesoderm becomes specialized and specified only when signals from neighboring cells activate a transcription factor called Twist. Twist then activates other genes that are necessary for mesoderm differentiation. Another example is the development of the eye in invertebrates. During eye development, signals from nearby tissue such as the optic vesicle are necessary for the specification for different cell types in the retina. Without these signals, the retina does not develop properly and the eye does not form. So next, gamete binding in sea urchin. Gamete binding is the process by which the sperm and egg cells recognize and bind to each other, leading to fertilization. The process of gamete binding has been extensively studied in sea urchins, which are commonly used as a model organism in developmental biology research. So here is an overview of the steps involved in gamete binding in sea urchins. First, chemotaxis. The sperm cells are attracted to the eggs by a chemical signal released by the egg cells, which is called chemoattractant. The chemoattractant is a peptide called ResAct, which is released from the jelly layer surrounding the egg. Then sperm-egg recognition. The sperm cells recognize and bind to the egg cells through specific molecules on their surfaces. In sea urchins, this recognition is mediated by the binding of the protein binding on the surface of the sperm to the receptor on the surface of the egg. Sperm-egg recognition. The sperm cells recognize and bind to the egg cells through specific molecules on their surfaces. In sea urchins, this recognition is mediated by the binding of the protein binding on the surface of sperm to the receptor on the surface of egg. Next, acrosome reaction. After binding to the egg, the sperm undergoes the acrosome reaction, which is a series of biochemical events that allows the sperm to penetrate the egg. During the acrosome reaction, the acrosome, which is a structure at the tip of the sperm, releases enzymes that break down the jelly layer surrounding the egg. Next, fusion. Once the sperm has penetrated the jelly layer and reaches the surface of the egg, it fuses with the egg cell membrane, leading to the entry of the sperm nucleus into the egg. Fertilization. The fusion of the sperm and egg nuclei leads to the formation of a zygote, which is the first cell of the developing embryo. The process of gamete binding in sea urchins is tightly regulated by various factors, including the chemoattractant released by the egg, the molecules on the surface of the sperm and egg, and the enzymes released during the acrosome reaction. These factors ensure that fertilization occurs only between sperm and eggs of the same species and that the process of fertilization is highly efficient. These topics are very important to provide insights into broader process of fertilization and early development in animals. Sperm and egg binding is the initial step of fertilization in which the sperm cells recognize and binds to the egg cells to initiate fertilization. The process of sperm and egg binding is a complex and highly regulated process that involves a series of molecular interactions between sperm and egg cells. So chemotaxis. Sperm cells are attracted to the egg cell through the release of chemoattractant, which is a chemical signal produced by the egg cell. In many species, the chemoattractant is a small peptide molecule released by the egg. The sperm cells follow the concentration gradient of the chemoattractant, which guides them towards the egg cell. Next, sperm-egg recognition. Once the sperm cells reach the egg cell, they recognize and bind to specific molecules on the surface of the egg cell. In most species, this recognition is mediated by the interaction between a protein molecule called bindin on the surface of the sperm and the receptor molecule on the surface of the egg. In some species, other molecules such as carbohydrates or glycoproteins may also be involved in sperm-egg recognition. Then acrosome reaction. After the sperm binds to the egg, it undergoes a process called the acrosome reaction, which involves the fusion of the acrosomal membrane of the sperm with the plasma membrane of the sperm. The acrosome is a specialized structure at the tip of the sperm that contains enzymes that help the sperm penetrate the protective layer surrounding the egg. During the acrosome reaction, these enzymes are released, allowing the sperms to break down the egg's protective barriers. Next, fusion. Once the sperm has penetrated the protective layer surrounding the egg, it fuses with the egg cell membrane. The fusion of the sperm and egg membrane allows the sperm to enter the egg cell and deliver its genetic material to the egg. Next, activation of the egg. The entry of the sperm into the egg triggers a series of events that activate the egg, leading to the initiation of embryonic development. These events include the release of calcium ions from the egg's endoplasmic reticulum, which triggers the fusion of the egg and sperm nuclei and formation of the zygote. Next, examples of species with different mechanisms of sperm and egg binding include sea urchin. In sea urchin, the sperm recognizes and binds to the egg through the interaction between the protein binding on the surface of the sperm and the receptor molecule on the surface of the egg. The acrosome reaction is triggered by the binding of the sperm to the egg, leading to the release of enzymes that break down the egg's protective jelly layer. Mammals. The process of sperm and egg binding is more complex and involves multiple molecules and interactions. The sperm recognizes and binds to the egg through the interaction between a protein molecule called LZUMO. LZUMO. It's a protein molecule on the surface of sperm and a receptor molecule called JUNO on the surface of egg. The acrosome reaction is also involved in mammals, but the enzymes released during the reaction helps to digest the proteins in the extracellular matrix surrounding the egg rather than egg cell membrane itself. JUNO is situated on the surface of egg. Next, epimorphic regeneration in salamander. Epimorphic regeneration is a type of tissue regeneration in which the lost tissue or organ is regenerated from a population of undifferentiated cells. Salamanders are well known for their ability to undergo epimorphic regeneration. Epimorphic regeneration is a type of tissue regeneration in which the lost tissue or organ is regenerated from a population of undifferentiated cells. Salamanders are well known for their ability to undergo epimorphic regeneration, which allows them to generate complex structures such as limbs, spinal cord, and heart tissue. So, injury and wound healing. The first step in epimorphic regeneration is the injury or amputation of the tissue or organ. In salamanders, the amputation of a limb or tail leads to the formation of a wound epithelium, which covers the amputation site and secretes a fibrin clot that helps to seal the wound. Then dedifferentiation. The next step in epimorphic regeneration is the dedifferentiation of the cells near the wound site. In salamanders, the dedifferentiation of the cell is triggered by signals from the wound epithelium and nearby nerves. The dedifferentiated cells become pluripotent, meaning they can differentiate into different cell types and form a mass of undifferentiated cells called the blastema. Then proliferation. Once the blastema has formed, the undifferentiated cells begin to proliferate rapidly, giving rise to a population of cells that can differentiate into different cell types needed for the regeneration of lost tissue or organ. Then redifferentiation and morphogenesis. The final step in epimorphic regeneration is the redifferentiation of the cells in the blastema into the different cell types needed for the regeneration of lost tissue or organ. The redifferentiated cells then undergo morphogenesis, or the process of organizing into the proper spatial arrangement to form the regenerated structure. Examples of structures that can be degenerated through epimorphic regeneration in salamanders include limbs. Salamanders can regenerate entire limbs, including bones, muscles, nerves, and blood vessels. The regeneration of the limb begins with the formation of a blastema at the site of amputation, which give rise to the different cell types needed for the regeneration of the limb. Then tail. Salamanders can also regenerate their tails, which contain spinal cord, muscle, bone, and skin. The regeneration of the tail begins with the formation of a blastema at the site of amputation, which gives rise to different cell types needed for the regeneration of the tail. Then heart tissue. Salamanders can also regenerate damaged heart tissue. The regeneration of the heart tissue involves the dedifferentiation and proliferation of the cells near the damaged area, followed by redifferentiation of the cells into the different cell types needed for the regeneration of the heart tissue. In summary, epimorphic regeneration in salamanders is a complex and highly regulated process that involves the dedifferentiation and proliferation of cells near the site of injury, followed by the redifferentiation and morphogenesis of the cells into the different cell types needed for the regeneration of the lost tissue or organ. Salamanders can regenerate complex structures such as limbs, tail, and heart tissue, making them an important model organism for studying tissue regeneration and development. Next, valval induction in C. elegans. Valval induction in Caenorhabditis elegans is a well-studied example of cell-cell communication during development. In C. elegans, the vulva is a multicellular organ that is essential for reproduction. Valval induction involves the coordinated signaling between two different cell types, the anchor cells and the valval precursor cells, to generate a functional vulva. Here is an overview of the major steps and parts that we will be learning tomorrow. Thank you for listening.